genes. We also ran this analysis through some gene ontology software, and we found that the top module here is associated with neuron differentiation, which is obviously quite interesting, especially because this is the dopamine metabolite network. So again, we're currently looking into this a little bit more further at the moment. So the third study I'm going to talk about, changing tact again, and this is really on functional studies and schizophrenia-associated genes. And this is work that my PhD student, Sam Washer, down here has been doing for his PhD, and he's just in his final year now. So as John's already sort of mentioned this project, and as he's also spoken about, though we have 108 loci for schizophrenia now, which is from about the 2014 paper. I think there's a few more with the new PGC analysis. But a lot of these are big loci with many genes, and we don't actually know what the genes are. So we kind of looked at the loci. We screened for loci that come up in our EWAS studies, because we want to use our own data. We looked at those that were had MQTL enrichment in both blood and brain. So we came down with 9 genes. We then looked to see if any of these had expression QTLs. We looked at gene function. We wanted them to be expressed in both fetal or neuronal expression, fetal brain and neuronal expression. Possibly had neuronal disease association, or had mouse knockout phenotypes that gave you some idea that might have something to do with brain function, and also looking at orthologs and zebrafish as well. So when we sort of did all this, we kind of came up with our favourite gene, which is ASVMT. And it's been associated with schizophrenia in GWAS, in EWAS and in TWAS. So it's obviously a fan of this. It's like the WAS gene, of schizophrenia. So there's this paper here that's particularly interesting, which is by Danny Weinberger's lab here. This one here, and they found that there was a specific isoform of this gene that was associated with schizophrenia. So what we did is took some of Eilish's work from a few years ago, and we sort of, where she really used this co-localization approach to sort of really home in on this region of this particular gene. So what does AS3MT do? Well, it catalyzes the transfer of a methyl group from SAM to trivalent arsenic. Basically, it plays a role in arsenic metabolism. It's got a full transcript here. There's also this other transcript, which is the one that's been associated with schizophrenia here. So so our hypothesis and our aims was that this particular gene has a role in neuronal development. It's expressed in developing brain, and which decreases postnatally, but it is expressed in adult brain as well. So our aim was to knock out this gene in neuronal cells using the CRISPR-Cas9 technology, and then examine the phenotype of the knockout and look at the transcriptional response to the knockout. So, as I'm sure you're aware, CRISPR-Cas9 is a fantastic sort of molecular toolkit that enables you really to sort of edit the genome. It's very simple to use. Any lab can do it, and it's quite a straightforward technology, very powerful. So we designed the CRISPR guide RNA to cover the methyl group, which is a functional domain of this particular protein, and it's present in both the normal AS3MT full transcript and also the transcript that is increased in schizophrenia. So we basically had 2 guide RNAs. The idea is to chop this bit out completely, so to create a deletion basically in this gene. So this is our sort of lab methodology. So we designed the guides, we phosphorylated anneals that had sticky ends, so we could clone them. We checked the plasmids using colony PCR and various different diagnostic digests and sequencing. We transfected into SHSY5Y cells, which are neuroblastoma cell line. Obviously, again, they're not ideal, but they're the sort of the most robust and easiest to grow neuronal-like cell line, and you can transfect them quite easily. There wasn't a lot of work, published data that people have used CRISPR on this cell line, so it was more of a sort of proof of principle that we could do this with the SHSY5 cell line. So we used electroporation for that. So what we did as well with our guide RNAs, we had attached different colours to the vectors. So we then did a FACS sort. So we FACS sort for cells that had both colours. So we knew that they had both guide RNAs. Therefore, they were most likely to have the deletion. We then after we FACS sorted them, we individually put each individual cell, and then we grew them monoclonally, which obviously took quite a few weeks. We then screened using PCR. So we this is just a PCR, and we did Sanger sequencing as well to confirm that we had the sequence disruption, because obviously we had deletion, so we were able to design a PCR that was able to that normally you wouldn't see because it would be too large. And we've also done Sanger sequencing as well to confirm they were there. We did a Western blot as well, which is a bit faint here, but you can see in the knockout, this is AS3T is in red here, and you can see it's there in the wild type. Ooh, gone. There we go. It's just here, and it's not present here, but you can see better in these pictures here, which we're able to quantify it as well. We also did some functional analysis. So what we did is you can actually differentiate SHSY5s using retinoic acid. So we did that for a week, over 7 days, and then we fixed them and then stained them for a neuronal marker, which is MAP2. We looked at neurite length, cell size, and cell number were calculated. We did see a significant change in cellular area. We didn't see any change in neurite length or neurite or total neurite length, but we're currently repeating this at the moment. I've got another student doing the same thing again, because these have been through the FACS, because obviously we wanted purified cell line for our transcriptional work, and we also done some demethyl work as well, which I'm not presenting here. But what we're doing now for the morphology is we're going to do the same CRISPR again, but rather than FACS sort and then clonally expand them, we're just going to, using the microscope, just measure those that have the fluorescent marker, so we know which one's which that way. So then we extracted RNA from these mutant cell lines and ran them through RNA-seq, and this is just our pipeline here. We use the DSeq2 software and features count. So what we were quite intrigued as well when we looked at the RNA-seq, so these are the knockouts here at the top, and this is our bit. You can actually see that this is the region here we knocked out and it's not there, but we do get transcripts of the other exons, which is interesting given that we don't actually get any protein. So this is just the top 500 most variable genes across samples, and DSeq2 found quite a lot of differential gene expression, and these are just some of the down and up regulated genes here, some of which are quite interesting. It's connectin, which is a member of the neurexin family, and there's MAOA here, which has obviously been associated with psychiatric disorders. So again, we're following up some of these at the moment. We also did WGCNA again, looking at the phenotype. We found 2 modules that were associated with the deletion that we created. So this is with the knockout. And in the turquoise module, the top one was cell adhesion. So with the student that's repeating this experiment now, what we're going to do is some cell adhesion experiments to see if there's any difference in cell adhesion. And the other module was DNA binding, was the top go term. So just to conclude, we successfully developed a CRISPR pipeline for knocking out specific regions of the genome in a neuropsychiatric relevant human cell line that's easy and cheap to grow. Unlike iPSCs, which the question was earlier, obviously they have their own issues as well. The knockout of AS3MT in the functional domain results in increased cellular length, but it doesn't change neurite number or length. We find over 700 genes are differentially expressed. And with WGCNA, we identified co-expressed pathways, and we found that these were involved in cellular adhesion and extracellular matrix formation, which we're going to investigate that phenotype further in further knockouts that we're doing. So the next little bit I'm going to talk about is work that's actually just started. This is what Sam's doing for the last bit of his PhD, and I've talked about using CRISPR to basically silence a gene, to knock it out, and look at the phenotype. But also, it can actually be used to alter DNA methylation at specific sites, which is obviously what we do in our lab. So this is an example of the epigenetic CRISPR, and we have these plasmids in our lab at the moment, and we're just trying to get it up and running. And what it does is the actual Cas9 has been mutated, so it can't cut anymore. So, but what it's fused to the Cas, you have either TET, which demethylates, or you could have DNT, which will methylate. So the idea is then, again, you use it in exactly the same way with the guide RNA, and it can actually methylate or demethylate a specific target. So we're using a lentivirus vectors. What we'll use is these vectors into HEC293, and this will be either with our guide RNA or virus with our DNT3A or TET1 attached. You also need enveloping and packaging because we're creating viruses here, and the HEC293 cells are really where to create the virus. So then we collect the media, and then we'll collect the viruses, and then we'll target our cell line. And we're going to start with SHSY5 just to see how that compares. We'll infect that with the viruses using lipofectamine, and then we'll use FACS again because again we have a fluorescent marker on these particular vectors, so we'll be able to FACS sort those that have the virus in. And with the guide RNA, we'll have a different colour, so we'll be able to FACS sort on the two colours, and we'll have SHSY5. We're then going to extract the DNA and RNA, and we'll do qPCR with target genes and also pyrosequence various different genes. Obviously, with epigenetics, you're altering the epigenetic. We don't know if it's going to be regulated, so you're not always sure which gene it's going to affect. So we might have to profile a number of genes in that region, and we might do some RNA-seq and things like that. But at the moment, this is currently ongoing in Exeter at the moment. So I've got my fingers crossed while I'm over here. Yeah, so just to acknowledge everyone over in Exeter, particularly Eilish as well, who's been involved with a lot of the data analysis. Essen is here, is the postdoc who's over in the Netherlands now. Sam's at the back, and then some other PhD students, Stefania, who's been doing a lot of the FACS work that John's already mentioned. So yeah, thanks for listening, and any questions?